I'm going to talk to you today about some of our data we generated looking at m6A mods in neuroblastoma. So just to give you some background on Epitranscriptomics, neuroblastoma, and nanopore. So Logan gave really good background to RNA modifications, but Epitranscriptomics describes the modification of RNA on transcriptomics. So RNA has more than 150 different modifications, and these play a vital role in RNA regulation, such as splicing, stability, and translation. N6-methyladenine, or m6A, is the most common modification of mRNA, and it's actually regulated via proteins categorized as readers, writers, and erasers. So you've over here in my figure, there's the writers, which catalyse the m6A mod on the mRNA. And MET-L3 plays a major part of that, along with some of the other supporting proteins. It's very context-specific, actually, what is kind of determines the fate of the mRNA with the readers. It's could be, it wholly depends on nuclear or cytoplasm, or it could be the DRAC motif site. It's a very complicated process. We've also got erasers, which are demethylases, such as FTO and ALK BH5. And most importantly, actually, often this process is deregulated in cancer. So neuroblastoma is a common childhood solid cancer with a diagnosis at a mean age of around 18 months old. It's a really heterogeneous clinical prognosis, so 50% of the patients actually spontaneously regress with limited to no therapy, and the other 50% actually are already diagnosed at an advanced stage with less than 40% survival rates. There are a lack of recurrent somatic alterations. There are some MICA and ALK P53 and TERT involvements, but it's not kind of across all of these patients. So finding novel treatments to treat the diverse biology in these high-risk neuroblastoma patients is really important and remains a significant challenge. Recent studies suggest that there is evidence that m6A modifications may play or be involved in neuroblastoma biology, susceptibility, and prognosis. So we can use Oxford Nanopore to study the epitranscriptome using this direct RNA sequencing to look at native RNA. When it's been really difficult in the past, Logan also gave a really nice introduction to this because as soon as we convert to cDNA, we lose all the RNA modification information. And some of the older technology is quite laborious and difficult to generate data. So Oxford Nanopore sequencing is actually the only technology where we're able to sequence native RNA. And we also get the transcriptomic information as well as the epitranscriptome. So for our study, we wanted to use this direct RNA sequencing to look at the epitranscriptome of high-risk neuroblastoma. So this is our workflow. Initially, we used the older chemistry, the RNA 002 direct RNA sequencing kits. So we grew 2 human high-risk neuroblastoma cell lines in the lab, this LAN1 and IMR32. We extracted with a trisol method to try and keep the DNA really nice and full length kind of transcripts. We put quite a lot of RNA into this prep, so 5 micrograms, but then split it across 4 flow cells on using the MinION flow cells on the GridION. We did canonical base call in a Minnow, used Minimap to align, and a nanopolish to event align, and then m6A net tool to identify the m6A modifications. So this is an example of one of our m6A mods that we found in this the 002 data. So just here at the bottom here, you can see this line here is one of the modifications we identified. You can also see it was only identified in a specific transcript. So it's actually an intron retention isoform, and the mod was only on that transcript. We did find more modifications, but so the tool m6A net we detected quite a lot of sites, but then actually when we filtered for high confidence, so we use the 0.9 cutoff, that reduces quite greatly the number of sites we identified. But actually when we looked at the genes that were shared between the two cell lines, so the ones that were modified, we found through a gene ontology analysis that there was biologically relevant processes such as histone H3K methylation. And this is actually really interesting because there's this kind of crossover, crosstalk with m6A regulation as well. So then at the end of last year, Nanopore announced and commercially released the 004 chemistry, which is really exciting. And we also had, in that meantime, got a Promethean 2 solo, this little guy at the bottom, which meant we could run the higher capacity Promethean flow cells. And we could also use Dorado now to look at the modified base calling model and then modkit to extract those sites from the merged BAM. So in this method, we actually use a different cell line. So we use the TH-MICN mouse model derived cell line, 9464D. The extraction was the same with Trisol. We put a lot less RNA into that prep, and it was just one flow cell. So this is actually really preliminary data. We've only just started to look at it, but I wanted to show you an example of actually the difference. One, we've got a lot more coverage across genes. This is a really interesting example, which is a MICN, and this is a MICN amplified cell line. So we get a lot of coverage over here, and we get a lot of novel transcripts that are highly modified. So over 88% of the transcripts actually have an m6A modification. So yeah, that's quite interesting, I think, observation, but we are still analysing that data at the moment. So just to kind of summarise what I've said today, Epitranscriptomics is a re-emerging field of genomics, and Nanopore offers us the opportunity to actually characterise the epitranscriptome. And we used the direct RNA sequencing kit on high-risk neuroblastoma cell lines to identify the m6A modifications. A gene ontology analysis suggests that there's an enrichment in biological processes, such as a histone H3K36 methylation. And we're actually doing further analysis using other RNA modification tools, as well as the 004 chemistry. But further analysis is needed to understand what the significance of these RNA modifications is to neuroblastoma biology. So I'd just like to say thank you to our funders, the Little Princess Trust and CRUK, as well as my colleagues Jane and Juliet, and Tom, who grew the cell lines in the lab for us. Thank you.